proteins everywhere because, as my fellow students in AP Bio know, proteins really are everywhere when you're talking about life. So to give some background, what are proteins? Proteins are biomolecules that are made of amino acids. And to just give a sense of scale here, cells are really small. Like, you need a microscope with 100 times to 400 times magnification to even see the cells. But even if I took a cell and blew it up to be about the size of this entire room, a single protein would still be smaller than a pea. And yet, they do all of these functions in our cells. They're essential for receiving and transmitting information. They're what drive your immune system. They catalyze reactions to photosynthesis and cellular respiration. They provide structure, like your nails and hair are all made of protein. And they're also what diseases are made out of course. And what's interesting about this is that these functions are all determined by the structures of the protein. As you can see, proteins have really I find them really beautiful and insanely intricate structures. And this is one of the appeals of biochemistry for me. It's the idea that even if you're looking at these tiny, tiny, tiny scales, you can find beauty. Like in the ATP synthase, this is an insanely complex molecule. And yet, it's something that you need for something as basic as just getting energy from your food. And also, I like that you can make some really crazy complexes between lots of small different proteins. Like this viral capsid has 60 different monomers all together fitting into a perfect 3D shape. And on to my internship. I interned with Professor Adrian from the UCSD SCAS School of Pharmacy and Pharmaceutical Sciences. She earned her Master's of Science and PhD in Computer Science from Tomsk State University in Russia. Then in about 2000-2001, she moved to the US here with her family, and she found herself out of work. And then she signed up to, in all places, work at the Scripps Institute for biochemistry, and she was working as a computational biologist. And now, around 10 years later, she's the head of her own lab. And I find it really inspiring that she was able to go into this field where she had no formal training. I took a look at her thesis for her PhD and her master's, and it's some very abstract cellular automata, which is a specific type of algorithm in computer science. And she had no training in biology, and yet she was able to go into this and learn everything on the job, and now she's the head of this group that not only does computational work, but is constantly doing experiments with chemokine receptors and GPCRs in general. And moving on to the internship itself, this internship was a computational chemistry internship, which I chose after I took AP chemistry, and I realized that I had I had never thought I would be a chemist because I'm afraid of chemicals. I used to be afraid of chemicals. If I'm at a gas station and I get a whiff of the smell of gasoline, I'll get paranoid about getting cancer. So I'll try not to breathe it in. But even then, I was like, well, this is actually interesting, so I guess I'm gonna try it out and see how it goes. And before the internship started, I got in contact with my mentor over email, and I asked her what I needed to do to prepare. So she sent me a list of articles to read with fun titles like Navigating Biological Space to Predict Polypharmacology, Off-Targeting Selectivity, and more and more and more and more articles I had to read through. This was an article that was written by my mentor and one of her students, which was really instrumental in learning my work. And as I was reading these, it's pretty tough because they use really technical language, and I had to have the paper in my hand and Google open in the other, and Google pretty much every single other word just to have an idea of what was going on. And then I arrived for the first day at my internship. It's at the SCAGS building, which I think is a really pretty building. So seeing the outside of this building was like really new and nice and neat. And going in, I also had to get this badge because there's a lot of biological agents. There's which actually was pretty bad for me because also scary diseases are something I don't like either. And every single door in this place is labeled with a big biohazard sign, which was pretty nerve-wracking for me going in. But then I went in and I found something quite a bit more human. It's a bit of controlled chaos in there. There's all sorts of machines everywhere. There's plastic pipette tips lying around. There's, at one point, I walked past the fume hood, and I looked inside the fume hood, and I saw there was a cardboard box with a paper napkin draped over it, with a poorly sharpied skull and crossbones on it, because it contained phenol, apparently, which I'm pretty sure is a carcinogen. And that's my workstation over there. And I was right next to all that going around, so I had people running experiments right next to me. And but I was working on the computer almost the entire time. So these are the other members of the Kufreva lab. There's Kriti Chahal, Sushini Soroy, Wei Junju, Tony Ngo, and Irina Kufreva. I was mostly, right now I'm collaborating with Wei Jun over the summer, and I'm also collaborating right now with my mentor. And over the summer I shadowed Kirti as she was doing some experiments. And of course I was working with Dr. Kufreva the entire time. And when I arrived, my mentor sat me down and gave me an idea of what I would be doing. And she pretty much told me what I would be doing is this: protein binding pocket structure comparison. So when you look at protein receptors, which are proteins that detect molecules like hormones, or for example, all your smell detection is based on smells binding to these receptors. And as you can see, there's a pocket right there. And the ligand has to fit into this pocket, and that's what allows binding to happen. And after binding occurs, it can either activate the protein or it can inhibit the protein. So this is really important in the process of cell signaling, regulating how cells grow, and etc. And so the idea is, if you can compare the structures of these pockets, if you have similar structures of pockets, then similar chemicals will bind to those pockets. And she told me that I would be using Molsoft's ICM script to do the analysis, along with a bit of Bash to automate. And I would be sourcing these 3D structures from the Capstone, which was a database that my mentor developed. And here's just an example of one of these 3D structures. This is the CXCR4 protein. This is actually a structure that my mentor helped resolve. And as you can see, the pocket, and you have a hormone, or I think this is some sort of peptide, as a small molecule. They all fit snugly into the pocket. And that just goes to show how important the structure of these pockets is in determining what will or will not bind to them. And so I sat down and for 6 weeks was programming. That was pretty much the only thing I was doing the entire time. So I got to stare at a screen that looked like this. I'm sorry, it's kind of, can't see very well, but looked kind of like this. Just writing lines of code after line of code after line of code. And that was an interesting experience, because going into this, I wasn't sure if I would maybe be, like, what this coding process would be like. I had coded in the past, but never something of this scale. I ended up writing between, I want to say, like 1,500 and 2,000 lines of code spread over 5 or 6 or so files. So managing that was an interesting experience, because I'd only written small pet projects with like 100, 200 lines of code. So this was definitely an entirely new scale that I'd never seen before. And this is ICM. This is the actual program that we use to visualize the proteins. I have it pulled up here on my laptop, so if you guys want to pass it around, and you can drag around the little structure and take a look at it. And this is what the results of my program looks like. So you have the light blue protein is one protein, and then the white protein is another protein, and it kind of overlays the pockets, and it gives you visual representation of how similar these pockets are. And after around 4 weeks of After about 4 weeks of full-time work, I got to this moment. And it might not seem like much, it's just me hitting the enter key, but this is me running the program against the entire database for the first time. As you can see, it says left 3,300 entries. I had to process over this absolutely massive database. The first time this program ran, it took like 3 hours to run. And then after that, it was 2 weeks of just me working to get it to run faster and faster and faster until eventually now it runs in around 30 to 45 minutes. And this is what the output of the program looks like. So you have, on the bottom left, proteins that aren't similar. On the top right, proteins that are similar. So this here is CCR2. I actually found some proteins that are similar, which we then tested to see if they were similar. Unfortunately, we got some preliminary results saying that they were similar, but after confirmation, we found out they weren't, which was a bit disappointing. But at the same time, it was really cool to see that this program I had written was actually experimentally verified, which eventually led to me being able to make this poster. I'm very proud of this poster. I have the PDF if anybody wants to see it, but I won't go too depth into it, but it has a nice explanation with diagrams of the more particulars of the method I programmed. And I thought at this point I was done, but then in March, my mentor invited me to go back into the lab and keep on working. That started when she invited me to go to this lecture by the Nobel Prize winner, Dr. Lefkowitz. He discovered the class of proteins, GPCRs, that we work with mainly in the lab. And it was really fun to get him to see him describe how he went through this process of discovery. And that leads me to the next project that I undertook, which is Boolean modeling of signaling networks. So proteins in cells, they have lots of interactions between proteins. And these interactions is what lets when you have a certain smell that comes into your nose, a ton of reactions happen, and that eventually leads to a signal somewhere else. Or for example, this network is for if a certain chemical appeared somewhere down here, it eventually goes through this network and leads to the cell moving. And you can model these networks using what's called a Boolean network. Each of these nodes represents either a state that the cell is in or a reaction that occurs. And they're all on or off, as you can see here. And then with that, you can take steps and simulate whether these will be turned on or off. And that gives you a simulation that more or less reflects what happens in reality. And my task is to take these simulation results and compare them to experimental results and see how accurate they are, which I'm doing by programming R. This is something I wrote this Monday to do some processing of the data files. And I'm actually going to be going back today to keep working on that because there's a grant proposal coming up in a couple weeks. And this is something that's going to be going in there. So I'm collaborating with Wei Jun especially and the rest of the lab to work on this project. And moving on to my ISO application. So the first application is of a lifelong self-directed learner. I had to be learning the entire time. There was no moment I could stop learning. From the very first moment when I had all these articles to read, and to even just learning how to program in these new languages that I hadn't programmed before, and also learning how to present this work in the poster. And even I thought that I wasn't done learning at the end because then I realized that I still have to keep reading articles to keep up. I thought that the 5 or so articles that I read over the summer were a lot, but then I would be sitting in a meeting with the whole lab, and I'd be discussing stuff, and it'd say like, "Oh yeah, did you guys see the Kellenberger 2008 paper about this protein or whatever?" And they're going back and forth, and just by mentioning a random article by the last name of the author and the year it was published, they immediately knew what they were talking about and were able to discuss it. So that really impressed on me how important it is to stay up to date with everything that's going on in your field. And the other ISO that I saw applied was that of being a collaborative individual. I had to collaborate, especially with my mentor and Wei Jun. It's been very interesting now that I'm working on the project with Wei Jun. I have to be going back and forth with him a lot. There's always emails going back and forth. We have face-to-face meetings. On Tuesday, my mentor, Wei Jun, and we sat in a 3-hour meeting trying to work through some stuff because we didn't know how to solve a problem. So we all sat down together in a room and didn't leave until we figured out how to do that. And I also have been doing some phone calls as well with him. He's called me in the middle of school a couple of times to get updates on stuff. And I also got to witness the collaboration between two bigger groups. So Dr. Kufreva is working on this grant proposal with Dr. Handel's group, and I got to see how there was some discussion, even butting of heads during the presentation, how they would go about presenting the information in the grant, but eventually they were able to resolve it, and they came to a solution that everybody was happy with. As for goals, my goal is first to get a bachelor's degree, and I'm leaning towards MIT right now. I'm very happy that I got into MIT. Shout out to Henry, who also got accepted there. So obviously the first thing I need to do in this path is to get a bachelor's, and I recently found out that MIT has a joint major in computer science and molecular biology, which just so happens to be exactly what I did and exactly what I like to do. And what is also offered there is a one-year extension to turn that into a master's degree as well. So I will likely be pursuing that. As for my work goals, they're kind of one and the same as my academic goals, because during these 4 or 5 years that I'm going to be doing my undergraduate work, I will be looking for a position as an undergraduate researcher in someone's lab. Because something I very much want to do is getting more on the experimental side of biochemistry. I only ever got to do a couple small things of taking care of a cell culture, but I never really got to do any real experiments. And that's something that still very much interests me, because it's just kind of a whole part of biochemistry that I haven't had the chance to even look at yet. And what that will also enable me to do is to get into a good PhD program, because I want to keep doing research. And to do that, I'm going to need a doctorate degree. If I eventually, whether it is I end up going into research or academia, I'm going to need some sort of PhD to be able to break into that field. So in conclusion, I want to just give one lesson. I know Karen already kind of covered this, but don't be afraid to ask questions. Even though I walked into this internship really nervous and expecting to fail, at the same time I had this attitude of mind that, oh, I can take care of everything by myself and I don't need help. And within the first couple of weeks, I realized that was not going to work. There was one time, for example, that I was writing some code, and every single time I ran it, the protein display on my screen, it would fly around the screen like spaghetti. And I sat there for an hour trying to figure out why this was happening. And I could not figure it out until I finally decided to shoot an email to my mentor, and she came over and told me, see, that line of code you're running right there, don't do that. And I removed that line of code, and I went about it a different way, and the issue was resolved. After that, I started going to my mentor more often, because I realized that if I'm able to collaborate with other people and admit that I need their help, I'm going to be able to work much, much better than otherwise if I was just going by myself. And in the end, this has just been an amazing experience. It's really let me know that biochemistry is a field I want to go into, and that I want to keep doing research, because that's just my passion. And on that note of not being afraid to ask questions, thank you, and if you have any questions.
Okay, well, anyone have questions? Do you know until when you're gonna keep working at the lab?
I'm probably going to keep working through the end of the school year. I'm actually looking to get another position at UCSD in Dr. Balatori's lab. There's an open, have you heard of that? Has Dr. Till told you about that position? You would probably be interested in that. That's more on the experimental side, so I'm looking to apply for that. But I'm probably going to keep working with Dr. Kufreva as long as I can, because I just really enjoy the projects we're doing.
What's been the hardest thing for you as you've been working in this?
Dealing with setbacks and when you get stuck. Because especially with programming, you just get to some points where you don't know how to tackle a program. And you'll sit there staring at a screen for 2 hours trying to think your way out of it, and you just can't. And that, in part, was just ended up taking determination on my part to keep working through, and also learning to go ask for help and get someone else's perspective on this problem really helped solving that.
So you already shared some advice on like what you felt like were important lessons, but if you were to like redo this whole experience again, and like what advice would you give yourself to have it run more smoothly? I would say, I guess.
Implement version control. So this is more of a programming thing, but as you're programming, you have your files and you're updating them and you're making new versions. I did not start out with a correct version control system like Git or CVS. I just had folders, version 1, version 2, version 3, version... Don't do that. It's a really bad idea, and it leads to a lot of messy code. I would also advise set backups, because once I made the mistake of running a command that deleted all of my code and about 4 days worth of work, and I just lucked out that it just so happened that my mentor had automatic backups running on the computer. But if I hadn't had that, I would have lost, not 4 days, but I wouldn't say at least like 6 or 7 hours of work, which would have been very disappointing.
Well, you did find yourself in those situations where like you're stuck for like over 2 hours, or like something just doesn't get unstuck, right? What did you do in order to like find a solution to that?
So I ended up having several methods. One was just taking a step back, taking a break, let myself rest for a little. That helped. Drawing diagrams in my lab notebook. I ended up keeping a notebook where I would draw stuff out that helped me plan stuff out better beforehand. But definitely the best way was just bouncing ideas off of people. I bounced ideas off my mentor. Sometimes when I got home to my dad, I'd ask him, because he's a math programming guy, so he knows how to tackle some algorithmic stuff, and that was probably the most helpful in getting past those frustrations.
What advice would you have for, say, someone who plans to major in comp sci that doesn't have as much like programming experience?
Okay, several. Again, learn to use version control. Comment your code, because on some of my earlier code, I made a function and then it worked, so I didn't have to come back for it to check it out. But then afterward, I realized it was really slow, so I had to go back and improve it. But it was just this 200 lines of code, no comments, and it's just kind of like, what was I thinking when I did this? And I can't tell what I was thinking when I did this because I didn't leave notes for myself. So comment your code well, and this is something I don't have a skill personally yet, but learn how to properly format your code and separate stuff into functions and different files, and use good practices, and that's something that I still haven't learned personally.
I think all of those tools that you've learned through your coding experience is going to help you in your wet lab experience when you eventually get there. I think it's really important to make really good lab notes, and I think that's going to help you overcome those uncomforts that you might have with your smells and the biohazard scare. So I think that's going to help a lot as well. I definitely encourage you to take on more experience in that, because that's going to help you. You've learned a lot, you know a lot. I can tell just by your presentation that you've studied this, you know it, you're comfortable with it, and so I'm really impressed with that. So good job on that. What did you learn about yourself in this process?
About myself. I learned that I was able to dedicate myself to projects for this long because before this I have an unfortunate tendency to like pick up hobbies up and to just kind of drop them after a while. But I learned that if there's a project that I'm passionate about and I have some structure to, I'm going to be able to stick with it.
I see just through your presentation today that you have grown up a lot since I had you when you were a sophomore. Another thing that I noted is that in the ISOs, you also gained humility. And through this process, you talked a lot about ask a lot of questions and be okay with like letting your pride go and be like, hey, I need help with this code. I don't understand what's going on here. So I really commend you for that, because that's going to help carry you a long way, especially when you go to MIT and all the successes you'll have there. So good job, Adrian.
Mm.